Hi, I'm Dr. Marc Rothenberg here at Cincinnati Children's with the Cincinnati Center for Eosinophilic Diseases, and I'm very pleased to share with you the research highlights from the last year. Just going to briefly review some of the things that I consider to be the most fundamental, thought-provoking new research findings that we have. Let's start off with the first slide, which is basically reviewing a recent article that we just published in the Lancet Gastroenterology journal. This was a critical study, involved a number of years of research testing a fundamental hypothesis, and the title of this study is shown here: benralizumab for eosinophilic gastritis, a single-site randomized double-blind placebo-controlled phase 2 trial. And what we showed in this study was that we could deplete eosinophils from humans, from patients with eosinophilic gastritis, and I call that the human eosinophil knockout experiment. We used the drug, it was very safe and effective, and we tested whether or not eosinophils, which are a hallmark of this disease, would actually be clinically beneficial when they were removed. And lo and behold, as we expected, we eliminated eosinophils from patients in a safe manner, but we found that this did not have a clinical benefit. So the histological, structural, endoscopic features, as well as the clinical symptoms as measured by a clinical score, did not improve, even though eosinophils were removed from the body. This is actually paradigm-shifting results because they changed the dogma in the field. Even though these diseases are called eosinophil-associated GI diseases, eosinophils actually may not be the culprit cell. And this is really setting the stage now for reconsidering how we diagnose the disease, how we monitor the disease, and how we treat it. More results are ongoing in terms of understanding what the next step is. These are really unexpected findings. Here at Cincinnati Children's, though, we've taken multiple approaches to understanding and treating these diseases, not just focused on eosinophils. And indeed, what I'll show you next is that we have a number of other promising avenues for treating patients, understanding the disease, besides just eliminating eosinophils. And this was another study that we just reported in September of this year, which was on the cover of this journal called Journal of Clinical Investigation Insight. What we looked at here is a discovery that we have, which is involving a series of proteins called the mini chromosome maintenance complex, which we showed drives the disease. This is schematically shown here, where we can see the eosinophil esophagitis schematic diagram. These are the epithelial cells which are expanding in the patients, something called basal zone hyperplasia. And when we block the mini chromosomal maintenance complex, MCM, with a particular drug, actually an antibiotic called ciprofloxacin, we can see normalization of the epithelial hyperplasia. We're able to do this in vitro, but also in vivo, in a murine model of eosinophil disease. This is very exciting because it's a new pathway, it's a new way of inhibiting eosinophilic esophagitis associated features that we think are clinically important, not focused on eosinophils, and we're using a drug, ciprofloxacin, which is often thought to be an antibiotic, but we're able to use it specifically for eosinophilic esophagitis, both systemically, more importantly, in a topical format. So I want to finally share with you other findings that we have focused on another pathway that we think is germane, and that's called the thymic stromal lymphopoietin, or the TSLP protein, which is a cytokine or an immune hormone that regulates type 2 immunity, which is related to eosinophilic esophagitis. And we've shown through a number of experiments that TSLP is germane for eosinophilic esophagitis based on genetic predisposition. But also in this particular study, we changed the way we view TSLP, not only driving the innate immune system, which is eosinophils and epithelial cell changes, but the fundamental cornerstone of the immune system, which is the adaptive immune system, which is the T cell, the memory T cell that's being driven by the food antigens. We're showing in this particular study that TSLP receptors are expressed by these pathogenic T cells in the esophagus, and that this actually is an activating factor for T cells. It causes changing in the chromatin overexpression and transcriptional activation of critical mediators, including the type 2 cytokines, which are made by the Th2 cell, like IL-4, IL-13, and IL-5 and IL-3, that are targeting the allergic inflammatory cells and feeding back on the Th2 cell, driving their proliferation. So these are exciting findings also because there are drugs that block TSLP that are now being used in clinical studies and being further considered for eosinophilic esophagitis based on this. So this final figure kind of summarizes where we stand today based on our research, understanding the mechanisms of EOE, which we think are driven by genetic factors, which are genetic variations in the chromosome of 2p23, which encodes for a particular gene called calpain-14. This regulates the epithelium itself in terms of the integrity of the epithelium, how it's held together. When we have a breakdown in the barrier, this could be inherited or due to various exposures in the esophagus, we have liberation of the TSLP under the genetic influence of a particular variant of chromosome 5q22. And that leads to the production of TSLP as well as its related cytokine IL-33 that target Th2 cells. And these Th2 cells make IL-13, which then stimulate and activate the epithelium to only make the calpain-14, which is the protease that we've shown is important in maintaining the integrity, also in the genetic control, but also down-regulating the integrity of the epithelium through a process called desmosomal adherence, which are down-regulated like this particular molecule called desmoglein-1. Now we know that this is important because IL-13 also drives eotaxin-3, which is an eosinophil chemotactic factor, and this is why the eosinophils accumulate. Through genetic rare occurrences in humans, very rare genetic disorders, or actually Mendelian inherited disorders, we know that this is important because we can see loss of function mutations in these desmosomal genes that can be associated with very high penetrance of eosinophilic esophagitis, and there are other ways that we've been able to see this as well. So there's a number of lines of evidence that we're accumulating through scientific experiments supporting this and introducing different ways of blocking the disease. We certainly know that blocking IL-13 now with the drug dupilumab is effective. We think that this TSLP will be important, and that protecting the barrier through other pathways is going to be critical in maintaining homeostasis and helping patients. So I want to close by thanking all the contributors to the research. Certainly this is a team that's very strong here at Cincinnati Children's. We've got a core group of the lab that's run by various managers, and we have a partnership with GI and Allergy, research coordinators that make sure that patients are enrolled in these studies, and the research lab itself. I want to thank all of you for watching this, but all of those that support our research, the patients that participate in the studies, and also the foundations, which include the NIH and the 3 institutes that are our main founders, the Food Allergy Fund that has been recently donating to us, David Denise Bunning and Amy and Michael Zuccarello. I also want to thank the Cured Foundation. This is the campaign urging research for eosinophilic diseases, which my lab is named after. I want to particularly thank Ellen Kodroff and her team for being super supportive of our research, allowing us to leverage the support from the NIH to do other experiments. And I want to also acknowledge the Buckeye Foundation, the Bornstein Family Foundation, the Louis B. Mayer Foundation, the Sebastian and Koulis families as well. Thank you very much for your attention today, and I hope that this was informative for you.